Question 1. Enzymes mainly function by Option A. Increasing product amount. Option B. Lowering activation energy. Option C. Changing cell pH. Option D. Slowing reactions.
Question 2. Enzymes are primarily made of? Option A. Carbohydrates. Option B. Lipids. Option C. Proteins. Option D. Minerals.
Question 3. Enzymes act on specific molecules called? Option A. Catalysts. Option B. Ligands. Option C. Substrates. Option D. Cofactors.
Question 4. Enzymes speed reactions because they Option A. Increase temperature. Option B. Provide energy. Option C. Lower activation energy. Option D. Alter substrate.
Question 5. Which statement is true about enzymes? Option A. Used up in reaction. Option B. Work as biological catalysts. Option C. Change permanently. Option D. Stop metabolism.
Question 6. Enzymes are highly Option A. Random. Option B. Specific. Option C. Neutral. Option D. Unstable.
Question 7. Enzymes usually work best at? Option A. Extreme temperatures. Option B. Neutral pH. Option C. Very acidic pH. Option D. Very alkaline pH.
Question 8. Enzymes speed up reactions by? Option A. Changing products. Option B. Increasing activation energy. Option C. Making new molecules. Option D. Lowering activation energy.
Question 9. Most enzymes stop functioning at high temperature due to? Option A. Activation. Option B. Denaturation. Option C. Hydration. Option D. Oxidation.
Question 10. Enzymes work within? Option A. Wide pH range. Option B. Narrow pH range. Option C. No pH effect. Option D. Only acidic range.
Question 11. The region where substrate binds is? Option A. Binding pocket. Option B. Active site. Option C. Allosteric site. Option D. Core region.
Question 12. Enzyme-substrate complex forms at? Option A. Active site. Option B. Allosteric site. Option C. Core. Option D. Surface. Question 13. Which model states substrate fits exactly? Option A. Lock and key. Option B. Fluid mosaic. Option C. Induced fit. Option D. Allosteric model.
Question 14. Which model states enzyme changes shape? Option A. Lock and key. Option B. Induced fit. Option C. Fluid mosaic. Option D. Chain model.
Question 15. Active site shape is determined by? Option A. RNA. Option B. Temperature only. Option C. Protein folding. Option D. Enzyme color.
Question 16. high temperature causes? Option A, activation. Option B, denaturation. Option C, division. Option D, stability.
Question 17. Optimum pH means? Option A, pH with fastest activity. Option B, most acidic pH. Option C, most alkaline pH. Option D, neutral only.
Question 18. Enzyme activity increases with substrate until Option A, saturation. Option B, denaturation. Option C, blocking. Option D, osmosis.
Question 19. Very low temperature makes enzymes. Option A, inactive temporarily. Option B, destroyed. Option C, highly active. Option D, toxic.
Question 20. Heavy metals act as? Option A, activators. Option B, inhibitors. Option C, products. Option D, buffers.
Question 21. Organic cofactors are called? Option A, metals. Option B, vitamins. Option C, coenzymes. Option D, proteins.
Question 22. Non-competitive inhibitor binds at? Option A, active site. Option B, allosteric site. Option C, substrate. Option D, cofactor site.
Question 23. Competitive inhibitor resembles? Option A, product. Option B, enzyme. Option C, substrate. Option D, cofactor. Question 24. Cofactors help enzymes to? Option A, stop reactions. Option B, work properly. Option C, destroy substrate. Option D, change pH.
Question 25. Which vitamin acts as coenzyme? Option A, vitamin C. Option B, vitamin A. Option C, vitamin K. Option D, vitamin B complex.